The next speaker will be Carl A. Michal. So Dr. Michal is a professor at the Department of Physics and Astronomy at the University of British Columbia. He completed a PhD in physics at Cornell University and proceeded to do a postdoc at the National Institute of Health. His research interests are focused on applications and instrumentation for NMR and MRI, and include the development of low-cost devices for NMR and MRI at low and high field, optical pumping in semiconductors, and understanding structure and dynamics in a wide variety of materials. His current research focuses on understanding and developing contrast mechanisms for the study of myelin in vivo. And today we'll give a talk titled The Physical Mechanisms of Inhomogeneous Magnetization Transfer. So yeah, thank you for being here, Dr. Michal, and you can have the floor now.
Thanks. Share my screen. Okay. So thank you very much for the invitation. It looks like a great meeting. Whoops. So yes, the title of the talk is The Physical Mechanisms of Inhomogeneous Magnetization Transfer. So first of all, what is ihMT? So it is what I would say is a clever variation on the much older technique of magnetization transfer, and it appears to be very sensitive to myelin. So magnetization transfer is, I guess one might say, sort of notoriously insensitive to specifics, right? It is sensitive to enough different things that it's hard to be terribly specific. But ihMT seems to be quite sensitive to myelin specifically. Myelin, of course, is the lipid-rich multilamellar sheath that insulates axons and accelerates nerve impulse propagation. I'm not going to say a lot about myelin because I guess I assume that most everybody here has some understanding. But I will turn to NMR in white matter. Okay? And if we were to collect an NMR spectrum in white matter, the signal has two main components to it, right? There's this large aqueous signal from the water, from the aqueous components, but there's another important contribution from the protons attached to molecules that are not aqueous, to the semi-solid components. And so this would be mostly lipids and proteins in the myelin and elsewhere in the tissue. These two components of the NMR signal are very different from each other, right? So the aqueous component gives a very sharp peak in the NMR spectrum, and so that would have a line width of something in the order of between 1 and maybe 50 hertz. So in the time domain, that's a signal that persists in the free induction decay for something greater than 10, maybe up to a couple hundred milliseconds. The signals from the lipids and the proteins decays much more quickly. So those signals disappear on the order of 10 to 50 microseconds. And so that corresponds to a line width in the spectrum, something like 10 kilohertz or so. And so these two very different components that we see. So the reason the signals from the semi-solid components disappear so quickly has to do with dipolar couplings. Okay, and if we think about the proton spins, each of those spins is associated with a magnetic moment. And you can think about the magnetic fields that come from these magnetic moments as the sort of typical dipolar pattern. And so from the point of view of a spin, say over here, it sees a magnetic field contribution from this other spin that's given by this formula here, where there's an orientation dependence and a distance dependence. And so this angle theta is the angle between the magnetic field, the main magnetic field, and the internuclear vector, and r, of course, is the distance between the two spins. And so this dipolar coupling is sensitive to the orientation and the distance between the spins. So water molecules, of course, tumble isotropically and diffuse rapidly, and so these dipolar couplings are averaged to 0. In the semi-solid contribution, semi-solid components of the tissue, every nucleus experiences a different environment. It sees a different combination of partially averaged couplings. So if we look at one spin here, and you look at its neighbors, it has some distribution of neighbors that are spin up and spin down at different distances and different orientations. But if you look at a different spin, it's going to see some different environment. And so all of these spins see different local fields. And that's what gives rise to this much broader distribution of resonant frequencies and this much faster decay in the time domain of the NMR signals. So now in the lipids, the lipid molecules, of course, rotate rapidly about their long axis, and they diffuse within the plane of the membrane. So those motions do partially average these dipolar couplings, but they're not sufficient to completely average them away the way the isotropic diffusion and motion of the water molecules of the aqueous components do. So there are dipolar couplings left in the lipids. So, okay, let's talk back about the magnetization transfer experiment. So the way a magnetization transfer experiment works is, from the NMR perspective, if you look at this as an NMR experiment and not so much as an MRI experiment, our NMR spectrum, again, has this aqueous component and the non-aqueous component. We're going to start off now by applying a pre-saturation pulse that's off resonance that hits just the semi-solid component, but is far enough away from the aqueous line that it doesn't affect it directly. So this is a low-power off-resonance pulse. And it has the effect of suppressing, of causing a partial saturation of the semi-solid NMR signal. If it's on for long enough, there'll be a cross-relaxation or magnetization transfer process that also suppresses the size of the aqueous NMR signal. And so you can detect this magnetization transfer by looking at the reduction in the signal from the aqueous components when you apply this off-resonance pulse to the semi-solid components. And so that's a magnetization transfer experiment. And the simplest way to quantify that is to look at this quantity called the magnetization transfer ratio, which is just the difference in signal with and without this off-resonance pre-pulse divided by the signal you see without the pre-pulse. So you get sort of a fractional reduction in the signal due to the pre-pulse. The ihMT experiment, as I said, is a sort of a clever variation of this. So now instead of doing 2 experiments, we're going to do 4 experiments. The first 2 are the same. The first is with no pre-pulse. We look at a spectrum or an image. The second is, again, the same, where we apply a pre-pulse to one side off resonance. The third experiment, we're going to apply the pre-pulse to the other side. And so this is sort of the same as the second experiment, but it's going to help take care of any magnetization transfer asymmetry. The fourth is the interesting one. So the fourth experiment, instead of applying the pre-pulse to one side or the other, we're going to split that pre-pulse up and apply it to both sides at the same time, but keep the RF power in the pre-pulse constant. So it's the same RF power as in the single-sided pre-pulse. And what's found is that there is a greater reduction in the signal seen with this dual offset pre-pulse than there is with the single offset pre-pulse under some conditions. Okay, so that's the ihMT experiment. This, again, the simplest way to quantify this is with this quantity called the ihMT ratio. And this now is the difference in signals seen with the dual pre-pulse compared to the single pre-pulse. Okay, and so this ihMTR is the fractional difference in the signal seen with the dual versus the single. I've divided this by a factor of 2 here to account for the fact that we're doing sort of a double difference, that we have we're subtracting off twice the difference because we're averaging over the 2 single-sided experiments. Most authors actually don't include the 2 in the denominator here, which I think is kind of a mistake, but something you have to watch out for in the literature. Okay, so experimentally, this experiment appears to be quite specific to lipids. And so this was one of the first, probably the first the paper anyway, on this subject that was published. And so there's a series of different phantoms here. The first row of phantoms is a cross-linked protein. The second is a set of, it's actually hair conditioner, which is one of the favorite phantoms for myelin, where the magnetization transfer effects are actually very similar as they are in myelin. And then there's a third row here you can barely see, that's our little vials of saline. And in the magnetization transfer experiment, you can see that both the protein and the myelin show up quite similarly. In the ihMT experiment, though, the hair conditioner / myelin shows up while the protein kind of disappears. And so this was also demonstrated in vivo. As a nod to the theme of the conference here and the question of, is this a quantitative technique? There is actually some work out there now that does make it appear as though this technique can be quite quantitative and sensitive to myelin. And so these are experiments done in genetically modified mice that produce green fluorescent protein in their mature oligodendrocytes and is believed to be a good marker for myelin. And you can see there's a very high correlation between the ihMT ratio and the green fluorescence. Okay, and these are all different variations of the ihMT experiment tuned in slightly different ways. But you can see for each of these experiments, there does seem to be quite a nice correlation between the green fluorescence signal and the ihMT ratio. One thing that's worth noting here is that each of these graphs has a non-zero intercept, so that even when there is no green fluorescence signal, which would indicate no myelin, there is still some ihMT. But there is a nice linear correlation in most of those cases. So I want to focus on how does this experiment work. So the simplest kind of a system to think about what's going on in this experiment is a pair of coupled protons. So from a quantum mechanics point of view, a pair of coupled protons forms a three-level system. Okay, there actually is a fourth level, but that doesn't participate in the magnetic resonance. So this pair of coupled protons produces an NMR spectrum that has two lines in it. The two lines are separated by twice the dipolar coupling strength. And I'm going to look at sort of diagrams like this, where I have these three levels. And the filled circles here are trying to communicate the populations in each of these different levels. And so these you should take as sort of the difference from thermal equilibrium, or from equal populations in thermal equilibrium. And so the lowest energy level has the greatest population, and the highest energy level has the lowest population. And then the transitions here are shifted slightly by the dipolar couplings. Okay, so the ihMT experiment, we're going to walk through a version of the ihMT experiment applied to this simple spin 1 system. So if I collect a spectrum of this system in thermal equilibrium, I'm going to see these two lines. And the amplitudes of the lines you can think about as coming from the population difference between the two adjacent levels. So in this case, each of these population differences is different by 2, right? There's 4 spins in this state, 2 spins in this state, so there's a difference of 2. And here there's a difference of 2. So the total Z magnetization available in this system, in this state, is 4. Now, I'm going to change my ihMT experiment just a little bit, and I'm going to apply a selective pi pulse, and I'm going to invert one of the transitions or the other. So I'm going to apply a selective pi pulse here that swaps the populations of these two lower states. And so now if I go back and look, what's the Z magnetization available here to create signals? Well, I've inverted these. So where I did have a population difference of 2, now I have a population difference of -2. But in this upper transition, where there was a population difference of 2 before, now the population difference is 4. So if I add those together, the total Z magnetization is now 2 instead of 4. I can do the same thing to the other transition, and it works out the same. But now, if I take my selective pi pulse and I split it up so that I apply it to both sides simultaneously, my selective pi pulse actually becomes a pi over 2 pulse. And that has the effect of sort of scrambling the magnetization amongst all of these different states. And there's basically no Z magnetization left at all. So there'd be no signal at all after that selective pi pulse. And so if I look at what's the ihMT ratio, I can look at what's the difference in signal produced in each of these different variations of the experiment. And I can plug that all into this ihMT ratio formula, and I find it's a half. Okay, so in this simple spin 1 system where I have 2 protons, this ihMT experiment done in this way with selective pi pulses, or a selective pi over 2 pulse in the case of the dual irradiation, I see a large ihMT signal. Okay, so that's sort of the simplest way to understand what's happening in this experiment. Of course, the brain tissue is a lot more complicated than just spin 1 pairs, or pairs of protons. But if we start off by thinking about the lipids being composed of mostly methylene groups in the tail of the lipid, you can think about a methylene group as being two protons that are reasonably strongly coupled together with weaker couplings to other protons nearby. So we'll start with that picture and then add in couplings between the methylene groups in the lipid tail. And there's going to be some distribution of the strength of the couplings depending on what position in the lipid molecule, where in the bilayer this methylene sits, and then these couplings to neighboring protons as well. So that's going to give some broadening to the pair of lines we looked at. And then in any real tissue, there's going to be some distribution of orientations in any voxel you might look at. And when you do an orientational average over all possible orientations, you end up with something that actually looks quite complex. Well, looks quite simple actually, but it's what's known as a super-Lorentzian line that does have sort of a sharp peak to it, but has quite broad tails that are again on the order of this 10 kilohertz we talked about earlier. So the spectrum that comes out of lipids in actual tissue is actually a lot more complicated than just the spin 1 model would suggest. And so instead of thinking about this from the point of view of the quantum mechanics of just the single pair of proton spins, we want to think about this from a thermodynamic point of view. And it turns out this kind of a system you can think about as consisting of two relatively separate thermodynamic pools. And the first of these thermodynamic pools is probably what you're familiar with thinking about in terms of spins in a magnetic field. And so this is what we're going to call the Zeeman reservoir, where basically the energy associated with the Zeeman reservoir is measured just by how many spins are pointed along or anti-parallel to the main magnetic field. The second of these thermodynamic reservoirs is the dipolar reservoir. And the sort of measure of the order, the energy in the dipolar reservoir really has to do with how many spins are aligned in the fields of their neighbors. And in the high temperature approximation, these two reservoirs are actually independent of each other, right? That you can flip one spin to change the energy in the Zeeman reservoir without that affecting the dipolar reservoir, or you can do a flip-flop in the dipolar, a flip-flop of two spins, which will change the dipolar energy, but not change the Zeeman energy. And so these are two sort of different sort of figures of merit of these two different systems that have to do with the same set of spins, but different aspects of them, of different aspects of how they're ordered. Okay, and again, the Zeeman energy has to do with how many spins are just aligned or anti-aligned with the magnetic field, and the dipolar reservoirs, how many spins are aligned in the fields of their neighbors. And so if we think about these two different reservoirs, right, each of them can have a temperature, okay, and the temperature, again, is related to these quantities we just talked about. There's another temperature in the system, and that's the lattice temperature. And so the lattice temperature is what you probably think of as the temperature, right? This has to do with molecular motion, how fast molecules are tumbling and diffusing and wiggling and all the rest of that. And then we have these relaxation processes, okay? So the things that what brings these other thermodynamic pools into thermal equilibrium with the lattice, right? For the Zeeman reservoir, it's T1, okay, and I'm going to call it T1Z for just to be specific to the Zeeman reservoir. But there's also a T1D, right? This dipolar T1, which tells you how long it takes for this dipolar reservoir to come into thermal equilibrium with the lattice. And these two different T1s, they may be sort of similar or they may be quite different depending on what the molecular dynamics look like in the system. Now, normally those two reservoirs don't talk to each other directly, but when an off-resonance excitation is applied, one effect of that is actually to have these two subsystems communicate with each other. And so when you apply this off-resonance excitation, as you do in the magnetization transfer experiment, magnetization flows basically from the Zeeman reservoir to the dipolar reservoir and back again. However, and if T1D is long, they will share the magnetization, right? The two pools will sort of share the magnetization. However, this dual frequency irradiation, if you apply the RF pulse, the pre-pulse, to both sides at the same time, this coupling between the reservoirs doesn't happen. And instead, all that happens is a suppression, right? This partial saturation of the magnetization. So the single-sided and the dual-sided irradiation actually do quite different things here. So again, the double-sided irradiation just saturates the magnetization. The single-sided irradiation connects these two reservoirs and leads to the sharing of magnetization between them. And the result of that is that the attenuation of the signal is less in the single-sided irradiation case. And that's the case where you find this ihMT ratio greater than 0. Now, there's another important piece to this. In some circumstances, you don't see an ihMT ratio greater than 0, even if there is a dipolar reservoir. Okay, and so these systems of the cross-linked protein we saw earlier, the ihMT ratio is just about 0, and there's lots of dipolar couplings in the proteins there. And so that has to do with the time scale of T1D. Now, if T1D is very, very short, then the magnetization that is shared from the Zeeman reservoir to the dipolar reservoir never comes back, right? Then that magnetization is basically leaked out to T1D relaxation to the lattice, and it doesn't come back to the Zeeman reservoir. And so in that case, the single-sided irradiation produces pretty much the same attenuation of the signal as the dual frequency irradiation. So this experiment is sensitive to the T1D of the dipolar reservoir. Now, there's a set of equations that can be used to describe this, okay, it's called the Provatorov equations, and this matrix rho here is a bit of a weird thing. It's got two components in it, which are basically the Zeeman, the order or the magnetization in the Z component of the magnetization is the first component. The second component in this matrix is the dipolar order. And so the off-diagonal elements here represent communication between these two reservoirs. And so for single-sided irradiation, you can see that there's a communication between the reservoirs given by this transfer rate omega, which is set by the dipolar coupling strength. Okay. W here is the saturation rate. So this is how effective the RF is at saturating the magnetization. You can see that contributes to both of the Zeeman and the dipolar terms. With the double-sided irradiation, Provatorov theory shows that these off-diagonal elements go to 0, and so that communication between the two reservoirs is severed, it disappears. And all we see is attenuation of the magnetization, partial saturation. So the net result of all of this is that the ihMT effect depends on T1D. Pulse sequences can be tuned to select different ranges of T1D. There's been some very clever experiments by other groups to build pulse sequences that do these ihMT experiments in ways that can select for either sort of narrow ranges or broad ranges of T1D values. And there's potentially lots of interesting experiments that can be done with that. To talk a little bit about T1D and what does T1D depend on, turns out T1D depends on fluctuations in dipolar couplings. And I'll try to make this relatively quick. But if you write out the dipolar couplings between protons in its sort of full quantum mechanical glory, you get this these 6 terms that have spin operators in them and space operators, or these orientation dependencies. And okay, so the first term here doesn't actually cause relaxation, but the other 5 terms all cause relaxation. And they have different orientation dependencies, but the spin operators are different. And so these first 2 terms are single spin operators. Sorry, let me say it again. C and D are single spin operators. The B term is the flip-flop term. Okay, so this B term is responsible for one proton flipping up and another proton flipping down. And that term, right, is sensitive to molecular motions on the time scale of the inverse of the dipolar coupling strength. C and D are single spin flips, and those are sensitive to molecular motions on the time scale of the resonant frequency. And then these last 2 terms are double spin flips, where 2 protons both spin up or flip down. And that's sensitive to molecular motions on the time scale of twice the resonant frequency. And what's important here is that the first term affects T1D, but not the Zeeman T1. And all these other terms affect both the dipolar and the Zeeman reservoirs together. And so this ihMT experiment is sensitive to this B term that you can't see in ordinary relaxation experiments. The dipolar, right, this dipolar frequency, again, we said is on the order of tens of kilohertz or so, whereas these other terms are hundreds of megahertz, depending on what magnet you've got. Now, the motions in the lipids, right, so the fast rotation of the lipid molecules within the bilayer and the diffusion within the bilayer, those are actually faster than this tens of kilohertz time scale. And so the ihMT experiment is not really sensitive to those measurements so much, but it does seem to be sensitive to slower collective motions, so larger scale fluctuations of the membranes. And so it looks like this actually might be a really interesting and useful experiment to study, health of the membranes, because it is sort of sensitive to these larger scale collective motions. And with that, I will conclude that ihMT is a variant of magnetization transfer that is specific to tissues where aqueous magnetization exchanges with semi-solid components having sufficiently long T1D. Pulse sequences can be tuned to select for different values of T1D, and this T1D relaxation differs from T1 relaxation because it is also sensitive to slower collective motions of the membranes. I'd like to thank some of the students who've worked on ihMT-related experiments with me, Alan Manning, Luke Reynolds, and Sarah Morris. And thanks to some colleagues at UBC, especially Alex McKay, who's sort of got me interested in some of these things in the first place. And thank you very much for listening.
Thank you very much, Dr. Michal. That was a very, very informative talk. Chris, do we have any questions in the chat?
Sure. We have one. Can you talk about the sensitivity of ihMT to the integrity of the myelin sheath? So you mentioned it could be used to look at potentially membrane health. So what happens in the case potentially of myelin breakdown, or with aging, we see sort of the decompaction of myelin sheaths. So will that potentially also impact the ihMT signal as well?
So, I mean, this is fairly speculative, but I would think it should be. I would think that, if you have a decompaction or a breakdown in the health of the myelin, I would think that would cause some fairly substantial differences in the T1D, because there would be a lot more sort of large scale fluctuations in the membranes. And I think a single membrane versus the compact structure of myelin would give you quite different T1D values. And there's lots of work, I think, that needs to be done to really sort of verify that. But I would say it should be useful there.
Excellent. A couple more questions have come in. You mentioned ihMT requires a dipolar coupled spin reservoir. Do we know anything about the limit of the size of the reservoir that's needed? So how small is big enough that we'll be able to observe this ihMT effect?
Yeah, so I mean, the theory there shows that a single spin pair should work, right? A single spin pair is really all you need. You need to have dipolar couplings where there is a dipolar reservoir to allow this sort of flow back and forth of order between the Zeeman and the dipolar reservoirs, and a single spin pair is good enough.
Awesome. And one more from Seo Zhang. You sort of mentioned at the end that pulse sequences could be modified. So could you briefly speak on what ihMT requires for a pulse design? And how can this be implemented on conventional scanners? Yeah,
so I think I would say that if the scanner can do an MT experiment with just a pre-pulse before an image acquisition, it probably can do an ihMT experiment. The simplest pre-pulse that you can do for this dual system is where the pre-pulse is just modulated sinusoidally, which splits the excitation into two, right? So you apply either an off-resonance pre-pulse or an on-resonance pre-pulse that is sinusoidally modulated at the frequency, at the resonance offset you want. So that's the simplest way of doing the experiment conceptually. Some of these other techniques that are sort of more sensitive to particular ranges of T1D do something a little bit different, where They do a series of off-resonance pulses, and you can do selective off-resonance pulses either all at one frequency offset, or you can alternate them so they go back and forth between the two offset frequencies. And the timing delays between the sort of going back and forth, those are the sorts of experiments you can do that become sensitive then to particular T1D ranges, right? You can play with how much time there is between the two different offset pulses. And that's been shown to be really sensitive to T1D.
Excellent. Thank you.